Take out all reagents. Prepare 9060W plate. Pipette 100% ethanol. Isopropanol. Add 100% ethanol to washing buffer to concentrate according to the label of the reagent bottle. Then mark them. For convenience's sake, mark numbers for each column and row of 9060W plate. Add 300 L isopropanol per well in columns 1 and 7 of 9060W plate. Add 200 L lysis buffer per well in columns 1 and 7 of 9060W plate. Add 500 L washing buffer to 100% ethanol has been added per well in columns 3 and 9 of 9060W plate. Shake the bottle containing Magbead's suspension solution and vortex for 2 minutes to ensure that the Magbeads are fully resuspended before use. Add 10 liters Magbead's suspension solution per well in columns 3 and 9 of 9060W plate. Add 100 L RNA-free water per well in column 6 and 12 column of 9060W plate. In the biosafety cabinet, add 300 L samples per well in columns 1 and 7 of 9060W plate. Put the 96DW autoplate and 8-channel combs into the instrument and run the program. After 7 minutes, remove the 96DW autoplate. Transfer the samples from column 6 and 12 to the centrifuge tubes and store them